Good afternoon, everyone. So here we approach the special contribution session, and I'm very happy to introduce Professor Zukin from Nadia Clinic in Ukraine. For those who still don't know him, he's been on the news in many international channels recently about his achievements on pronuclear transfer that led to successful pregnancies. And he was so kind to accept our invitation, and we will be hearing his achievements live. And due to the time constraints, I can only have two quick questions at the end of the session. And please, Dr. Zukin.
Thank you. First of all, I would like to thank organizing committee, Professor Bakhchiji, and personally my old friend, Doktor Necati Fındıklı, for the invitation to be here. And I would like to present for your attention the topic about the first three parents' babies born to infertile couples, scientific and social aspects. I would like to say straightly that I think that the term three-parent baby, it's more like the journalist invention. But from another point of view, that everybody understand what has happened. And I think in the future we use the more scientific term cytoplasmic replacement. Yeah, just a moment, please. The problem of the cytoplasmic control for early embryologic division has been known many years ago. It is a publication in 1982 by the group with the participation very famous embryologic professor Wettingham about the cytoplasmic control for implantation development in vitro in the mouse. And in this very old publication, they received the special inbred line of the mice, which has been infertile, and after injection of the cytoplasmic from another source, they could receive the pedigree from this infertile line. It was a lot of investigation in the mouse about the transfer of cytoplasmic, and I think that one of the key points of this investigation, it was the works of very famous Professor Jacques Cohen in the end of 90s and beginning of 2000s about the so-called cytoplasmic transfer, and his data is summarized in this slide, and you could see that they make 37 cycles, including 33 patients. They receive 70 babies born, 12 deliveries, and they have the early XO miscarriage and late termination with XO. And after the few years, it was investigation about the existence of donor mitochondria, and it was confirmed in these two babies born in this area. And after this, it was the document from the American Regulating Authority, FDA, that formally forbid any investigation for cytoplasmic transfer in the United States. But in spite of this, in 2003 it was published by a group of Professor Zhang, the case about the pregnancy with a human nuclear transfer. And in this case, it is very interesting that the first publication has been made in 2003. It was like abstract in ISRM meeting. But the full presentation has been made in 2016, and it is a short description of their job. This has been performed in 2003, 30 years old patient, 2 prior attempts, normal responder, complex 2 cell blocks in both attempts. They make the pronuclear transfer. Most embryos pass the block. From the 9 eggs, they receive the 5 2 days embryos. Transfer 5 embryos, receive triplets. they make selective reduction, but unfortunately the pregnancy has been terminated in 92 weeks, and both of babies were died. And it is a publication which has been published after many years. But in the October of 2016, it was published new case by this group, and it was the case of spindle nuclear transfer for mitochondrial disease prevention for the Lyme disease. And this publication has been made in all newspapers over the world because it is the really first so-called three-parent baby. And in April number of Reproductive Medicine Online, it was published the full publication of the group of Zank about all details of this, I think, that historical investigation. For this moment, we know different type of nuclear transfer. GV transfer, just a moment, please. GV transfer, spindle transfer, pronucleus transfer, and polar body transfer. It's 4 types. Main indication for the using of the different type of nuclear transfer is prevention of mitochondrial disease, avoiding of so-called embryo arrest. And very shortly about the mitochondrial disease, that after the investigation of the Human Genome Project, it is known that mitochondrial genome consists from 37 genes, and it is known a lot of diseases which transmitted through the mutation in the mitochondrial genes. And according to the last investigation, we know that for the PGD, the critical threshold is 18% of the mutant allele to avoid the transfer of these embryos. That which option could we propose for the family with mitochondrial disease? You know that adoption, oocyte donation, prenatal diagnostic, preimplantation genetic diagnosis, and from the 2060 nuclear transfer. But the preimplantation genetic diagnosis for mitochondrial disease has definite limitation. First of all, that some kind of so-called homoplastic mutation, it's not could give you the suitable for embryo transfer embryos. And the typical example of this is the Leber atrophy of the nervus oculus. Not always possible to find mutation-free embryos, and level of heteroplasmy in embryos could be different from the clinical significance threshold in baby born. That when we make the preimplantation genetic diagnosis, it is very difficult to predict we transfer the healthy or the sick embryos. And it is maybe the first example of misdiagnosis in the preimplantation genetic diagnosis of mitochondrial disease. The group, Nathan Treff, very famous group, they make the preimplantation genetic diagnosis in the cases of confirmed of so-called Melas syndrome, and they transfer the embryos with the level of the mutant allele only 12%. But after this, after the baby was born, it was the publication about the clinical manifestation of the symptoms of this syndrome in this baby, and the level of mutant gene in different tissue is different, and even in urine that it is more precise place for detecting of the, it was 42%. And it is one example of the limitation of preimplantation genetic diagnosis of mitochondrial disease. And for this moment, the main question is how is it safe and precise preimplantation genetic diagnosis? Could we avoid transmission of mitochondrial diseases by nuclear transfer technique? And what is the risk of segregation, development of mutated genes during the baby development? It is a publication that has been made in the ISRM meeting, and a lot of clinical background for explanation how we could manage the pronuclear transfer has been made by the group from Newcastle University, by the history Professor Turnbull. And my conclusion for today is that nuclear transfer is a powerful tool for preventing of mitochondrial diseases. But in our case, we use another indication, the so-called embryo arrest. Why did we decide to use pronuclear transfer for patients with embryo arrest? First of all, we hypothesized that embryo arrest could be connected with some unknown cytoplasmic factors. And I would like to remind you, the first publication of Professor Zhang has been made in 2003 in the same indications. We have animal models of embryo arrest in mice and bovine. It is already describes the human mutation with family infertility unknown etiology. And about the embryo arrest, it is not a lot of publication, and I think that this publication, which dated 2008, may be more detailed than we know for today. But during the last years, increased the interest for so-called human subcortical maternal complex genes. And the main candidates for pathology of early embryology development is these 4 genes. That in this, another, I think that very deep publication, it was analyzed the mutation and connection with embryologic picture in different species, including the man. And even they propose the so-called theoretical function of different genes and their development for early embryology development. And if you will see this publication, you could see the gene candidates for the early maternal effect genes in mammalian early embryo development. You can see that the list of genes, and from all of this list of genes, we will check. Only 2 genes has been confirmed that they influence for early embryo development in the human, because most data received in mouse and bovine. But according to the very deep investigation, this part of genome is very conservative. And I think if we will focus to investigate the patients with the early embryology, with disturbance of early embryology development, we could find the same mutation in human also. And 2 mutation is included now in online Mendelian inheritance in men. It's so-called pre-implantation embryonic lethality 1 and 2, and they connected 1 with gene TLE6 and the 2 with gene PADI6. And I would like to show you some example of the genes which found in the some disturbance of the early embryologic development and fertilization in human. It is publication from China, and they found the 10 patients with the different genes mutation in gene TUP8, and you could see that in all of this case they have very low fertilization, and very low cleavage rate without the blastulation. The same publication, more deep, and then from another China's group about the mutation in PADI6 genes. I think that it is very interesting case that this lady came in infertility clinic for infertility treatment. They make the few IVF cycle and receive very poor quality embryos, but in this situation, her parents has been cousin, brother and sister, consanguinity, and in this family they have she has 2 infertile sisters, 44 and 46 years, 1 fertile sister, and the 2 fertile brothers. It's a classical autosomal recessive inheritance, and in this situation they found the mutation in gene PADI6, and in some patient with only single cases, the family, they found the same. And the most interesting is the embryologic description and the picture of the development of embryos. You could see that for the embryologists, and understandable, that these poor quality embryos for day 3, we could discard from the embryo transfer according to the poor embryo quality. What's happened in this situation? The group Al-Azami, if I don't mistake, from Saudi Arabia, describe from the consanguinity couple, two daughters, two sisters, with a mutation in gen talia 6. All of these genes is included in the subcortical maternal complex, and I think that future investigation could show us the influence of gene mutation in this complex for the early embryonic development. And the simple thing, what did we do? That first of all, I would like to show you the animation which describes first of all, we fertilize simultaneously the patient egg and the donor egg. After this, we follow for the development of the pronucleus. Approximately simultaneously, we remove the pronucleus from the patient egg, from the zygote and from the donor zygote. Introduce in the donor enucleated egg the patient pronucleus. And after the same activation, we receive the normal embryo development. It is animation. How it looks in reality. Please open this file. It is the real picture under the microscope. It's removing of the pronucleus. All of this manipulation has been our embryologist Pavlo Mazur from our clinic. He was the first who could make this. And after this, the pronucleus introducing in enucleated donor egg. That's all. Okay, which result we receive? It's a general result from our first group. We divide all our patients for the two groups, under 37 and more than 37 years. In the group under 37, we have the 6 patients. We have the 35 reconstructed zygote, 21 blastocysts, and from this 21 blastocysts, 12 euploid blastocysts. We make 3 embryo transfer, achieve 3 pregnancies. For this reason, I don't calculate any percentage. And 2 deliveries and 1 ongoing pregnancy. In the group of the more than 37, we have the 9 patients, 58 reconstructed zygotes, 22 blastocysts, only 1 euploid embryos from this group, and this embryo transfer in this patient in the age of 49 has been unsuccessful. It is our primary results. Main conclusion from our first experience, pronucleus transfer could not overcome the aging aneuploidy. It's understandable. And for this moment, we stopped to include in this group the aging patients. It is a description of our case. Case 1, 34 years, more than 10 years infertile. She has multiple ovarian stimulation, but no embryos development. And in our cycle, we reconstitute 7 zygotes. 4 achieve the blastocyst stage. Trophiectomy has been provided in day 5. 3 embryos were uploaded. After the transfer of 1 euploid embryos, the normal delivery has been developed. And January 5, the healthy girl has been delivered. It is a confirmation from the German laboratory when we make during the pregnancy, we send the DNA from amniotic fluid cells to confirm the successful oocyte donation. In this table, you could see that it was investigate the haplotype of the fetus, mother, and mitochondrial dose. Its conclusion, that its confirmation that any mitochondrial haplotype of mothers could not be found, but paternity has been confirmed by this laboratory, Santo gene. That very important question, what is the level of the heteroplasmy in this case? Because this cytoplasm consists not only from the donor mitochondria and some part of mother's mitochondria, but in dimension which we make here, we know exactly that here the heteroplasmy less than 10%. We would like to investigate more carefully the baby to know that what is the level of the heteroplasmy, what is the part of the mother's mitochondria in this baby. And it is a follow-up of the baby in the age of 3 months. She came for me a few days ago. The baby is absolutely healthy. We did not find any problems. The second case, it is approximately the same. And for us, it will be very interesting to show the embryoscopic picture of this patient. Please open this. You could see that very well-known picture how the embryos develop. And in this case, we have the embryo arrest with very poor quality of embryos. I think that this next speaker could explain that it is very bad embryo development, Dr. Mezeger. You could see that in the time of normal blastulation, we have only the, the degenerated embryos. And I would like to repeat this picture. It is very similar for the picture which had been described in the PADI6 mutation by the Chinese group. And for this moment, we formulate the indication for the pronuclear transfer. Age before 37 years, normal ovarian reserve, excluding male factor, 3 and more unsuccessful cycles with the absence of blastulation, with very poor quality on day 3, and wish to avoid oocyte donation. Because for this patient, it's only 3 options, or to go for oocyte donation, or to become childless, or to use pronuclear transfer, at least to try. Main aims for future investigation. First of all, the main criticism is concerning the follow-up. And for this reason, we organize a special program of following up of this patient every 3 months to be sure that this patient are healthy. Finding more suitable, according to the indication, efficient procedure. You had that theoretically we could make by the different ways, GV, spindle, pronucleus. We could use for the nuclear fusion, electrofusion on Sendai virus. Nobody knows. And from my point of view, the main aim is to find the strict genetic marker of embryo arrest or pre-implantation embryonic lethality. For this moment, we are working together with the Chinese group from Fundach University, and I would like to show you some our results. It is a history of patients which came in our clinic a few years ago. She has 3 or 4 IVF cycles. We received 56 eggs and we did not receive any blastocyst. And pregnancy has been achieved in the first cycle with oocyte donation. We invited this patient for the group for pronuclear transfer, and during genetical testing in this Chinese laboratory, we found the TLE mutation in homozygotic status, the same as described by a group of Alazani, or it is a pre-implantation embryo lethality number 1, according to the OMIM online Mendelian inheritance. From my point of view, it's the second case describing. You could see that the same mutation, the same gene, but another mutation. Another patient who is preparing for you is a lady from Sweden. She has 7 unsuccessful ovarian cycle and 5 cancellation according to poor embryo quality and 2 embryo transfer on day 2. And for us, it is a very big challenge to help this couple to get the pregnancy, because we receive a lot of requests after our publication, and for this moment we try to find the right criteria how to help this couple. And main threshold for developing nuclear transfer technology. First of all, it's a legal restriction. You know that for this moment only one country, it is the United Kingdom, it's allowed, but according to the indication to avoiding of mitochondrial diseases. But my logic, it's very simple. To avoid mitochondrial disease, we could use oocyte donation. We try to this patient to have their own children. Was this patient with pre-implantation embryo lethality or embryo arrest, which is not describing very well, could not help have their own babies. Another and unknown long-term effects. Clear, I have read the book of one of the pioneer of the IVF, Robert Edwards, and about the 40 years, it was the same argument against the IVF. For this moment was born more than 6 million babies over the world. And how we will know if we will not do this? It's impossible. Attitude for patient for this technology, because even not all patients who has the indication for providing of this procedure, they agree for this procedure, they refuse. It is the right of the patients. Opinion of medical society, because in our clinic came a lot of patients from abroad, and when they said that they went for the pronuclear transfer in Ukraine, I heard from the patient the comments of the local doctors. You understand that? I understand that it is a beginning, but somebody must begin with this. And attitude of public authorities. Clear, I spoke with these 2 ladies which have the babies after the cytoplasmic replacement. And nobody would like that his baby will be named the 3-parent baby. Like said, the mother of our first baby said, it is my baby and my husband. I don't know her 3rd parent. I would like to thank for the collaboration John Zhang for inspiration, Takumi Katyusha for his belief, Mina Alikani and Jack for their kind support and advice, Professor of Group Li Wang from the Fudan University for genetical testing, that our embryology team, and especially Pavlo Mazur, he is here, and our genetical team, and all of our Nadia Clinic team, which take more or less participation in this. Nadia, it's meaning in Ukrainian language, hope. And I think that we will give hope for the hopeless couple. Thank you for your attention.
Thank you for this nice presentation, and also thank you for keeping up the time. So, as I said, I can take 2 quick questions. Is there any questions from the audience?
Doctor, thank you very much. As I understand from your slides, the main indication for your procedure, I mean the so-called pronuclear stage transfer, except mitochondrial DNA disease and the arrest of the embryo, right? Is that correct? Okay. And for this one, you are doing nuclear transfer. That means you are bypassing the mitochondria, the mother's cytoplasm, right?
Cytoplasmic replacement.
Yes. That means, could we say all of the cleavage stage embryo arrest, all of the reason coming from the mitochondria instead of the nuclears?
Yes, very good question. That, for example, the gene TLS6 is coded by the nucleus DNA. But he produced the protein which is located in cytoplasm. You understand?
No.
That we change the protein but not change the genes. And the participation of the genes in very early stage only through protein.
Where are the genes? Gene in the nucleus?
Gene in the nucleus.
Nucleus, yes.
Produce the protein.
Yeah.
The main dogma of molecular biology. Gene, DNA, RNA, protein. And this protein block, we don't know why, the early embryo development. And it is a lot of, for this, it's a lot of investigation of this in mammals.
That means you are targeting the cytoplasmic proteins instead of the genes, right?
Yeah.
All cytoplasm.
Yeah. So, I mean, the real problem still stays in the genome, but you just bypass the mechanism so that, you know, you can go on.
Just bypass, right?
Yeah, just bypass, yeah.
Thank you.
What I find challenging in this study is that people are talking about the pronuclear replacement for mitochondrial disease treatment. But what you actually aim is that you use the technology for infertility treatment.
This technology could be used for mitochondrial disease, avoiding of mitochondrial disease also. And clear the publication from the Newcastle, it's describing of this topic.
Yeah.
But a lot of questions concerning why in his first case, Dr. Zank used the spindle embryo transfer. He said that according to the ethical reason, the couple would not like to destroy the embryos. And if you took out the spindle and transfer the spindle, it is not the embryos. It is the main question.
Yeah. Okay. Another question? Okay. And thank you very much, Valery.
Thank you.
And so we can move to the next talk, and I'd like to invite the chair of the next section, Dr. Chiray.
Good afternoon, ladies and gentlemen. This is going to be the last session of the day. After this session, I'm hoping that we will get to know each other better. We will socialize, eat and drink again. This session, Dr. Marcus Messeker, I don't think he needs any introduction at all, but I will try to say a few words about it, if that's okay. So, Dr. Messeker is the scientific manager and statistical advisor of EV Equipo. Is that right? And senior embryologist EV Valencia. I know he's sitting on the editorial board of RBM Online and Fertility and Sterility, at least. He has published over 140 articles, 50 or more review articles, book chapters, so we know him very well. He's an associate professor in Valencia University. Yeah? He's sitting on many grants and paper awards, a lot of research success. His research interest is mainly on embryology and male infertility, although I have seen him this morning performing vitrification in the laboratory by his own very own hands. We know Marcus Messaker from time-lapse publications, mostly. He had been the pioneer, at least on the European side, of the time-lapse technology. So his talk today is time-lapse reloaded, full automation for embryo selection and improved designs.
Thank you very much for the introduction. And also we'd like to thank the organizer, especially Dr. Necati, for inviting me to come here to this wonderful island. Also, I've never been in the Turkish side, so I'm very excited with the idea to visit it. Well, the title of the lecture, as you have seen, is going to be about, what is potentially new in the technology, what else can add to the last publications in the field. And as always, try to start trying to underline that, the genetics of the embryos, the chromosome content is very important, and all of us are agree that if the embryos are genetically normal, we consider that healthy, but we must not miss that there are other factors that are potentially affecting the outcome, and I bring with me to this presentation this paper that was published by Jason Swain, between others, in which we try to review or underline the other factors that are potentially affecting the outcome, because the oocyte is a very large cell, and potentially these changes in the cytoplasm may affect the outcome. So trying to figure out how we can study the cytoplasm of the embryo, we came with the study of the cell kinetics, which is a non-invasive aspect of the embryo development that we can observe. And in the last years, many technologies related with this cell kinetics study has come to us into the market. So I have bring you here 5 of the most well-known, some of them that just recently came, and we have been lucky because we have been able to try in our lab. So I will show you a little bit of our experience with some of these machines. But definitely the introduction of all this technology in the market has allowed us to become friendly users and use and analyze and get a lot of experience and try to improve the knowledge in this technology. So since the technology came to us, we had the hypothesis that if we're able to do more observations of the embryos, potentially we may select better. And also, if we're able to give them better culture conditions because we keep in the incubator longer or we don't remove from the microscope, also we have the potential to keep in better development. So that hypothesis is possible by the introduction of these continuous monitoring system incubators or time-lapse incubators or cinematographic incubators. It depends the way that they would like to mention these instruments that are providing those aspects. So I will go through some examples, the last of the examples that we have done in research in that line. First of all, I would like to show you this study which is done by one of our embryologists, Ayla Coelho, which is going to be a speaker on Saturday, in which we just plan to analyze the behavior of the blastocyst after warming, because normally we do the warming of the blastocyst and we keep in the incubator for 2, 3, 4 hours, and then we can see how the blastocyst is doing the expansion, what was the initial size of the blastocyst, what was the final size, the thickness of the zona pellucida when we did the warming, or after 3, 4 hours of observations, and doing that with the time-lapse technology. So you can get a lot of nice data, interesting data, as you may see in the graphic, or in the tables. You can also compare if there is any difference between the blastocyst that implanted or not, depending on the behavior of that blastocyst after warming, regarding with the thickness of the zona pellucida or the initial area or the final area of the blastocyst, or even the inner cell mass area. So if you can take a look to this data, there are some of the behaviors, for example, the initial thickness of the zona pellucida or just the final expansion area of the blastocyst, that is significantly different between implanted and not implanted embryos. So there's room to do such a kind of a study in this way. So for example, if you split the maximum blastocyst area in 4 categories, you will see that the implantation potential may change. And even if we go to the initial area of the blastocyst and the final area, and we split in 2 categories depending on the diameter on the surface or the square, the micrometer squares, we can detect 2 types of blastocysts with different implantation potential. So this is a good example of that we are doing more observations of the blastocysts, that we can split the blastocysts in different categories depending on these sizes of the process of expansion, and that may be linked to the implantation. So what we did together with that was to analyze the behavior of the blastocyst. In around 10% of the cases, the blastocysts do not expand, and that lack of expansion is linked with a very low implantation potential. Also, we have seen that the embryos that finally implant tend to start the expansion sooner than the embryos that did not implant. So again, another feature that is potentially interesting for selection. So then we wonder, for selection or just for the knowledge of the future outcome of those blastocysts? We wonder, let's try to compare a way of evaluation of the blastocyst after warming, just measuring the areas and the thickness of the zona pellucida, with the standard morphology classification of the blastocyst following the Spanish Society of Embryology, and put all these together in a logistic regression analysis, together with the maternal age or together with the day of beatification. Well, if you take a look to the data, apart to the day of beatification that is affecting, and we have better results transferring on day 5 than in day 6 blastocyst, even being warmer blastocyst. If you take a look to the data, what we have seen is that if you are able to measure some parameters of the blastocyst, those are more relevant to detect the implantation potential than just taking a look to the morphology. So again, we demonstrate that doing more observations of the blastocyst even after warming, we can have a better idea of the future outcome. This work is now under publication in Fertility and Sterility, hopefully will appear this year. Another study that I think that was quite interesting by another of our embryologists, Raquel Herrer from IBI Zaragoza, was to analyze the presence of vacuoles in the embryos around embryo development, from the oocyte until blastocyst. Well, one of the things that we realize is that the appearance of vacuoles is not the same during all embryo development, and most of the vacuoles appear around the time of compaction and also in the time of the zygote formation, the time in which we're able to see the prenuclei. Together with that, and keeping in mind that the numbers still are not very big, and we hopefully will have more data in the near future, if you take a look to the embryos that were vacuolated or not, and of course by this way of observation in which we take a look to the embryos during around 5 days, we can detect better where are the vacuoles and when appear, we can see that the outcome of the embryos with the presence of vacuoles is different between those that has and not, with difference around 10–11%. Still, the numbers are not big enough to become significant, but it looks that there is some kind of difference in the behavior of the embryos having or not vacuoles. So, moving to another step, we can just do more observations, or even we can apply already existing algorithm like the EVA algorithm, that basically what is doing is just analyzing the first cleavages of embryo development and is classifying the embryos in 3 categories, high, medium, or low, and those categories are related with the blastocyst prediction potential. So that was the initial paper published by Joe Conahan like 3 years ago, and what we have done is try to validate this data by our own experience with retrospective analysis with a large data set of patients. And in this study, what we tried to see was quantify the effect of the selection by the EVA in comparison with the standard morphology of the embryo, and even the day of transfer, because we combined day 5 and day 3. Well, again, if you take a look to the data, when the machine is classifying your embryo as high, the implantation potential of that embryo is multiplied by more than 2.24. And if you put in the same model the morphology of the embryo became less relevant, or let's say that the classification of the embryo following the system, the EVA system, is more predictive or is stronger like a marker of embryo quality than the morphology of the embryo. So again, we have demonstrated that morphology works, but we can apply an alternative or an additional marker of embryo selection or of embryo quality by using this technology, and is able to distinguish between embryos with different implantation potential. If we go deep into the detail of the study, and just taking a look to another logistic regression analysis, doing a comparison of the cases in which we have a patient in which at least one of the embryos that we transfer, because sometimes we do one embryo transfer or two, single or double embryo transfer, is classified as high by the system, the chances to become pregnant, has an ongoing pregnancy, is multiplied by 2.5. And even, which is more interesting, if you take a look to the figure 2, is that when you have an embryo that has at least one high embryo, the chances of becoming pregnant is around 67%, and if the patient has no high embryos, it's only 46. But if you compare with the presence of a very good morphology embryo, quality A by our classification, or with a patient without any embryo with A quality, there's no difference in the pregnancy outcome. So again, we demonstrate that this is an independent parameter and can be used to improve the selection. This is a publication that was done in 2016 in Fertility and Sterility. So, what is coming from the observations of many patients, many embryos all around the world, and many publications quite interesting, is that we have detected very abnormal behaviors of the embryos, like direct cleavage, abnormal cleavage, reverse cleavage, multinucleation, that used to happen in a quite frequent session around embryo development and can be linked with the outcome. If we take a look to all those papers that presented this information about the presence of abnormal behavior, we have seen that in most of the studies, the incidence of abnormal cleavages or direct cleavages or abnormal behavior is quite high. It's between 15 and 30%. So keeping in mind that we have a very high proportion of abnormal behavior, we must take into account those embryos in order to take a look to the outcome. If you see the publications that were linked to the observation of this abnormal behavior and the outcome, you may see that in most of the cases, the implantation potential of those embryos with abnormal behavior is close to zero or extremely low. So really deserves to take a look to this data carefully and use those informations that has been published when we have this technology to avoid the transfer of those embryos like the first option. So then also the big question is, we have to do a randomized control trial, but the evidence that we have with the retrospective analysis probably is quite evident to avoid going through a randomized control trial in such a kind of observations for sure. But in any case, what is the information about the use of this technology, and if it's just providing better culture conditions, we may have an improved outcome? Well, there was an abstract presented last year in ESRE, in which they just compare the use of this technology of standard incubators in comparison with time-lapse incubators, just the undisturbed option with the disturbed one. And in that study that they presented last year, they reported an improvement in the live birth rate of around 10% and a decrease in the miscarriage rate of around 5%. So this was one of the first evidence reported about the use of this technology by a prospective way and just taking into account the culture conditions. And also, last year in ESRE, Markus Montag and Previnsky presented an abstract in which they summarized the 4 randomized control trials that are already done by using time-lapse technology plus algorithm of morphokinetics for selection in comparison with the conventional assessment. They put all together in this meta-analysis, and they end up with a conclusion of an odds ratio in favor of time-lapse technology in ongoing pregnancy, and also a decrease in the early pregnancy loss that is reported there in 1.5, the increase, and a decrease in the early pregnancy loss of around 0.66. So the evidence become quite relevant now, and just would like to underline one of the last papers that we are working with that now is in press in fertility and sterility, trying to study also the take-home baby rate, the delivery rate by using this technology, and also taking a look to the obstetrics and perinatal outcome, doing a comparison between the standard observations and time-lapse technology. And again, but when we did logistic regression analysis, we demonstrate that the application of this technology has provided us an improvement in the outcome in such a kind of studies, and improving the outcome in the take-home baby rate, which is calculated in 1.4 in the odds ratio. So what else we can do? Well, the future is coming basically with computers that may help us to improve the selection with another view of the embryo that we are not able to see with our standard observation. Let's say, take a look to the fragmentation patterns, the presence of some subcellular structures, the cellular shape dynamics, so many potential studies can be done. But definitely the time-lapse incubators has come with new incubators, a new era for the laboratory. Of course, behind all these new incubators, also the problems related with the cost, which are very high. We have been very lucky in the lab in the last years because we have had the chance to try one of the two new devices. One is the EmbryoScope Plus and the other is the Gary. We have been using still for a few months, so the clinical data that we have is still limited. But just showing you what we have seen new in this new technology in regarding of the EmbryoScope Plus. Well, a change with the standard EmbryoScope is that it has more capacity from 6 to 15 patients, and also another change that for us has been important, and in any case not very nice because we like to have the sample of the media of each embryo. All the embryos are cultured together in the same drop. So an increase in the capacity and a different way of doing the culture of the embryos, and maybe probably better observation of the embryos with better optics. Another incubator that we have been trying the last year, if this works. Well, finally, I would like to show a little bit of the results. This is just a civilian study that we did doing a comparison between the EmbryoScope Plus and the old EmbryoScope. The incubator, like itself, works very well, and the initial data was showing us even better blastulation, better viable blastocysts, and even better good quality blastocysts, but the numbers are still very low. So we will see in the next years what is coming. The other incubator that we are trying is the Gary. Well, Gary has come with a different concept because are different independent incubators with different cameras in comparison with the EmbryoScope, in which all the cameras were one camera only together and taking pictures of all the embryos just by moving around the embryo, so it's a different concept. We have been using also for a few months, so still the data that we have is quite limited. We're just testing the incubator, and as far as we have seen, our blastocyst rate, implantation rate, the cleavage rate or fertilization rate is more or less acceptable and is in the line of the other time-lapse incubators that we have been using. So what else is coming with the software? We have seen an evolution also in the software of time-lapse analysis. So I have bring you here the introduction of a new software for time-lapse analysis. This is called Dana, and the concept is quite interesting. What we do is just using two variables of time-lapse. In this case, we use the duration of the second cell cycle and the transition of from 3 to 4 cells, the synchrony of the cell cycle. So what we have done is to upload a lot of information of morphokinetics of many embryos that we transfer, some of them implanted, some of them not. So you will see in the graphic red spots and blue spots, and also some white circles, which are the embryos of a patient that I'm going to use the technology to select my embryos. So the software basically what it's doing is create a cloud of embryos in which we have a lot of information of morphokinetics and in which we are able to distinguish between embryos that implanted or not. Then I upload my patient into the system, and the system automatically is grading our embryos in relation with the similarities or the distance with those embryos that I'm going to select with the embryos that implanted from a group of patients that I am studying. So immediately the machine is ranking you the embryos automatically. It has the possibility of using several morphokinetic parameters, so we can play with some of them, and also with different outcomes, or even we can compare between different clinics. Well, this software we have been trying to validate in a prospective study that we're trying to present this year in ESRE. And this is a prospective study that we did in around 280 couples, around 400 embryos transferred, from them 261 with no implantation potential. And we just did annotations until T4, until 4 of cleavage. So if you take a look to the data of the Dana validation, here the embryos are split in 3 categories in relation with the distance of the embryos with the embryos that implanted. So we classify the embryos, those that are far away from the average of embryos that implanted and those that are closer. If you take a look to these 3 categories that we have generated, the embryos that were closer to the embryos that implanted had higher implantation potential than those that were far away from that. So we have let's say, calculate or generate distances that are related with the implantation. So the system was able to create embryos with different implantation potential automatically, taking a look to the distance. And also you take a look to the ongoing pregnancy when we have at least one embryo that has been classified with a distance of less than 0.4 from the cloud of embryos that implanted. The ongoing pregnancy rate is around 11 points higher than those cases in which do not find any patient with an embryo that has this quite similarity with the embryos that implanted. So again, it's another example of more observations, better selection, but also using an automatic software that is able to classify my embryos and help me to select which one to be transferred. So the last thing that I would like to mention is the possibility of combining the time-lapse technology with the measurement of the sperm culture media. So I have bring you here a couple of studies. This one that was published in 2016, last year, in Fertility and Sterility. The first author is Dr. Tejera from IBA Valencia. It's one of the work of his PhD thesis. And in this study, what we did was combining the measurement of the oxygen consumption, the use of oxygen by the embryo, together with the detection of the timings of cleavage by time-lapse technology. So that was the idea, doing simultaneously two aspects. First of all, we measure the oxygen consumption, and in second side, we are able to detect when the embryo is cleaving. So that study was relatively quite interesting because we were able to detect when the embryo were cleaving, and also at the same time we were measuring the amount of the oxygen that were used by the embryo. So we were able to figure out if there were any kind of change in the behavior of the respiration of the embryo when the embryo is cleaving. Also, we analyzed the oxygen consumption in the interface without cleavage. We did it in all the embryos, we did it in embryos that were transferred or discarded, and also we did it between embryos that implanted or not. So this was the device and the design of the study, that those wells were very small wells and very deep wells, so there was like a nanosensor that were going through the embryo and was able to detect a decrease in the oxygen concentration because the embryo is using oxygen. So there was creating like a slope that was able to detect it, and each embryo were cultured in these individual wells. Together with that, pictures every 10-15 minutes were taken. So at the end, we were able to take between 2 and 3 measurements per hour for each embryo, and also together with that time-lapse evaluation. So around 90 measurements per embryo during embryo development until day 3. We compare between embryos that implanted or not. Well, if you take a look to the behavior of those embryos, you have seen that every time that the embryo produces any kind of change in the evolution, the pronuclear formation, pronuclear fading, first cleavage, second one, 3, 4 cells, 5, etc., there was an increase or a peak in the oxygen consumption. That was one of the first observations that we realized. The second one was that there was a decrease in the amount of oxygen that the embryo were using with the development until day 3. If we compare between embryos that implanted or not, here the data was even more interesting. The embryos that finally implant, in average, presented wider peaks of oxygen consumption during cleavage than embryos that did not implant. So there was a different behavior of the embryos between embryos that implanted or not in the time of cytokinesis. If we're able to combine the time of cleavage with oxygen uptake, you see the oxygen uptake in the up of the slide for the timing of cleavage of 3 cells and the time of cleavage of 5. And below the implantation potential. In somehow you see a relationship between the amount of oxygen that the embryo is using and the implantation potential of those embryos, and also link it with the time in which the embryo cleaved. So it came to us, this information, just thinking that if we're able to combine both, we potentially may improve the selection. So what we did was a logistic regression analysis. We analyzed the timings of cleavages of the embryos until 5 cells, and also we measured the oxygen consumption. And we end up with this logistic regression analysis. We were able to combine both, and we realized that when the embryo has an increase in the amount of oxygen using of 1 femtomole, there is an increase in the implantation potential of 1.6, and also if the embryo cleaves, the first cleavage between the cleavage of 2 cells is between 24 and 28 hours, also this is linked with an increase in the implantation potential of 1.7. But the important thing of this figure, of this slide, is that we demonstrate by logistic regression that if we are able to combine both, we may be potentially able to select better by the combination of 2 different and independent parameters. So finally, one of the last research that we are doing now is also analyzing the spent culture media of the embryo by using this technology called TCL. This is thermochemiluminescence. Basically, what we are doing is collecting the sample of the embryo in which the embryo has been cultured for 5 days. Then we put in this machine, which basically what it's doing is heating the sample, evaporating the sample, and measuring, counting the electrons that are emitted by this sample because of this heating and also this vacuum effect. The signal that is provided by this media is related with the oxidation potential, which means that if we are able to detect a higher signal, means that media was less oxidized, and we are able to oxidize more together with those that has less. So we would like to try to link the relationship between the outcome of that embryo or the quality of that embryo and this implantation potential. Well, this is one of the first data that we have that we are going again to present this year in ESRE. We were able to see that depending on the signal, these are counts per second that we got from the culture media, there was a link with implantation potential. So those cases in which the media was giving us higher signal were linked with higher outcome of those embryos. Also, if you take a look to the average of the signal and compare between viable and non-viable embryos, the transfer and vitrified embryos that are good quality blastocysts presented higher signal that embryos that were discarded because they were not viable or they were just used for observation because they didn't have good quality at day 5. So there was a link between the quality of the embryos and the signal provided by the system or the oxidability of the system. And also another thing that we compare was the culture conditions in different incubators. We compared culturing the embryos all together in the EVA, culturing in the Gary, and culturing in the embryoscope. And we observed that the oxidation potential was different depending on the type of incubator that we were using. So again, we were just wondering that we may potentially use this technology to compare different culture conditions and also to use like a quality control. So just to end, try to bring you some take-home messages of the lecture of today. Definitely, I think that this technology is an objective one, and I'm completely sure that will improve our outcomes in assisted reproduction. We can apply different strategies in our lab to improve our objectivity in our practice. One is using these continuous embryo monitoring incubators, and this is basically by 2 main reasons, because we may provide better culture conditions to our embryos, and also we provide more observations, which means better selection. The embryo monitoring systems that are in the market now are in permanent evolution, and we have seen that future hardwares, because new devices are coming, and also mainly new softwares based on selection of the embryos are being developed, and we probably will be able to use in a very short period of time. I also believe that we may provide also additional parameters like oxygen consumption or thermochemiluminescence or maybe study proteins that may help us in that selection of the embryos. And again, a good message for the embryologists that we need more. We need more cook. We need user embryologists and maybe less food critics. I mean, clinicians without experience with this technology that basically are opinion leaders but are not using in the day by day. So we need more work, we need more embryologists working in this technology, and I'm sure that in the next years we'll come with quite interesting data. And that's all. Thank you very much for your attention.
Thank you very much, Dr. Necati. Excellent presentation. I will allow 1 or 2 quick questions. Before that, can I just clarify in one of your slides, I think it was your paper in person FNS, comparing conventional incubators versus time-lapse. Did you say the odds ratio for female neonates was 2.0?
Yeah, it was yes, it was an increase in the
What
females.
is that?
Maybe the morphokinetic selection is altering the sex ratio. We have another publication where we will demonstrate that there are different behavior between the embryos. So maybe it's just observed by chance, or maybe by doing such a kind of selection, we may alter the odds ratio.
That will not make people very happy. We should be... Okay, Necati had a question. Thank you.
For the time-lapse technology, I always concerned about the culture media because the embryo is spending like 4 or 5 days in the same culture media, and we're changing the media every month for different batches. So if you just want to compare different parameters, you have to standardize to make everything the same for the rest. But culture media, I think, the main confounder in this kind of research. Is Dana like a self-learning software, or you just have to type something and just give you some result and just pick the embryo?
The good point of Dana is that it's multidimensional. So I think that, we are still using at the very beginning, but will be a great tool for quality control and also to be able to overcome the difference between different laboratories and such. Because the point is, with that kind of technology, we're able to put together the data of many labs in a cloud. So I guess that we will be able to see in real time if the different cultural conditions that we have between the laboratories are really affecting or not on how we can manage that. We have not reached that point, but the potential of this technology is because of that, because we will put all the data together.
Yeah. So, hypothetically, can we use a kind of like mouse embryo assay for every month when we receive the culture media? Just, put 100 mice embryo in the embryoscope and just score them. And every month you can just, to compare whether there's any differences in the different time points. I don't know.
For sure, and one of the things that we are really interested is in using this technology for quality control, to be able to detect any problem sooner than when it's too late and our patients are losing their pregnancies. And I think also there's a lot of potential because that continuous monitoring of the embryos will help us to detect quicker than just with the standard observation such a kind of problems, for sure.
Any more questions from the public? No. You have the luxury of having access to more or less like every time-lapse equipment, what is available in the market now. Do you observe, like when you're predicting the algorithm for implantation, there is a machine-dependent algorithm, or is it a universal algorithm for all of the models?
Well, what I can tell you about the algorithm is the last publication that was published by a group of UK, that they put all these algorithms together, and they observe difference between them, and also they say that in fact they create their own algorithm to select because none of them were fitted. But I realized when I was reviewing the paper that the algorithm that we published in 2015, in which we put together like 6 or 7 clinics with a huge amount of data, were probably the one that were fitting better with them. So I think that the time of algorithm was like a startup of the technology, but with these 2 or 3 dimensional softwares, we will not use anymore the algorithm. We will rank the embryos by similarity.
Right.
So that was the first let's say, the first trial to try to select. But now I think we will no longer need that, but because once we were able to create this cloud, by the first time you will be able to go into the software and compare your embryos with a cloud of embryos that implanted, or even by yourself, the machine, by self-learning, will improve the abilities of outcome. So we will not probably need to be worried about using one algorithm or another. It will be more simple, like that.
Okay. If there is no more questions, then I'd like to thank you everybody, and I think this session is over now. Thank you.